You want to say anything about that?
Yes.
Okay.
Please note, next week's lab on cell memories, there are 3 experiments, and they run somewhat simultaneously, so it'll be very important that you understand what you're doing in each of them and coordinate carefully with your lab partner. This is one of the labs where having a good lab partner really makes a difference. Note that for experiment 2, which is on page 4 of the lab exercise, you have to develop an experimental design. Right? Each of you, not each pair, each student, and you write up that experimental design and hand it in beginning of class on Monday. And even if you have lab on Thursday, you're going to hand in that experimental design on Monday. Okay?
And we'll give them some time on Monday to work it out with their lab partner to refine the procedure.
It's supposed to happen in lab, but we could do that in class.
We could give them a few minutes during Monday's session. So really, you're going to need to bring 2 copies, bring 1 in to be graded, and then 1 to keep to share with your partner. I mean, this doesn't take a long time, but it requires you to read through the lab first. And then to think a little bit about what you did last semester in terms of developing a reasonable experiment.
As well as what you did in lab 1. Because what you did in lab 1 bears heavily on experimental design.
Yep. Okay, we'll also have our reading quiz on Monday. That will be, is it what, 5.1 through 5.4, is that correct? Okay, so it's not the entire chapter. So it's things we should have covered. We have an exam scheduled for the 29th. I know this sounds craziness to you, but it's not too early to be thinking about studying for that exam. What kind of things should you be doing? You should really be looking over your notes on a regular basis. And I consider the PowerPoint slides part of the notes, okay? The exams come from the notes. I can't reinforce that enough. If there is something that we did not talk about in class that is in the reading, I will not hold you responsible for that on the exam, okay? Other things you should be doing to prepare is you've got all those study guides. You should be writing out answers to those study guide questions. And I think it's important to actually write answers out as opposed to reading the question and answering it in your head. Because if you answer things in your head, you skip over the things that you are uncomfortable with. If you actually have to write out an answer, it helps to reinforce learning it. Another thing you could do and should do is, what if you write out a lousy answer and then that question appears on the exam and you write the same answer? You're not going to get all the points, right? So what you should do then is, once you've written out answers, check them with somebody. Bring them by my office, for example, and have me look them over. Bring them to a mentoring session and have Austin look them over and say, is this a good answer? Would this get me the full points on an exam? And then there are also 2 old exams that are posted on the website. You should be looking over those. That will give you an idea of the types of questions I ask, kind of the format of the exam. We'll talk about that a little bit on Monday next week. But you should be familiar with those particular exams. It's a new exam every time, but the general structure of the exam is pretty much the same every time. And if you go through, you'll see on the old exams that you should be able to answer the questions pretty much from the old exams from your PowerPoint notes and lecture notes. And you'll see that the essay questions oftentimes are adapted from, or even times verbatim, the study guide questions. Okay? So there shouldn't be any surprises in terms of kind of the nature of the exam and what it looks like. Okay? So get started early. I can't stress that enough. Okay, so we were talking about osmosis last time, and we will continue to elaborate on that a little bit in preparation for this week's lab coming up. A couple of criteria for osmosis. Number 1, we're only talking about the diffusion of water. So it's a special case of diffusion. You have to have a differentially permeable membrane involved for it to be osmosis. Osmosis is a type of passive transport. What does it mean when we say transport is passive? It happens without the input of energy. Another way to say that, it is spontaneous. Okay? And basically, it's obeying that law of thermodynamics that I mentioned, the second law of thermodynamics, which we will elaborate more on when we start talking about metabolism in a couple of weeks. But things diffuse because of the second law of thermodynamics. That's the bottom line. Well, from our consideration, what's really going to be important is, as a result of osmosis, cells can either gain water, lose water, or be in an equilibrium where they neither gain nor lose. And what ultimately does that depend upon? That really depends upon the type of environment that that cell is in. Okay? Cells, whether they're part of the body, whether they're a single-celled paramecium, live in a fluid or aqueous environment. Okay? The cells of your body are bathed in fluids that we call tissue fluid. And the nature of that particular fluid is really the determining factor about whether the cell gains water from the environment or loses water to the environment. What is it about the environment that is the critical determining factor? Solutes. Okay? What's a solute? It's something that dissolves in a solution, okay, and the solution is the water. What direction is water going to be moving in response to osmosis? Because this is a case of diffusion, what is the rule that determines which direction water moves? Where's it moving from? From water's higher concentration to water's lower concentration. Now, if I start throwing some things into water, some solutes into water, that dissolve in that solvent, is that going to affect the concentration of that water? Actually, it is. Yeah. If I've got a pure solution of just water, we consider that to be 100% water. Okay? If I throw a bunch of salt in there that dissolves, will the concentration of water still be 100%? No, some of that space of that volume is going to be taken up by salt molecules as opposed to water molecules. So if I throw enough solute in there, then it might be a 50% water solution. Okay? So the addition of solute to the solvent lowers the concentration of the water. The more dissolved solute that there's present in that solution, the less the water concentration is. Well, there's three terms we're going to be dealing with that determine or describe the tendency of one solution to gain or lose water to another. And those particular terms are hypertonic, hypotonic, what's the third one? Isotonic. Okay? What does the prefix hyper mean? If somebody is hyperactive. Excessive or a lot. Okay? What does hypo mean? If you have a hypodermic syringe, what does it do? It injects below or beneath the skin. Hypo means lower than or below. What does iso mean? Equal. Okay? These particular terms are relative terms, and they compare one solution to another. What are the two solutions that we typically are comparing? Typically, we're comparing the environment that the cell's in, the water that the cell's floating around in, if you like, and what's the second solution? The cytoplasm. Okay? So, if I've got a cytoplasm that contains 30% solute, and I put that into a solution that contains 50% solute, which of those solutions is hypertonic compared to the other? The environment is hypertonic compared to the cell, right? What is the cell compared to the environment? It is hypotonic. So you see how these terms are relative? Okay. Well, if we're talking about pure water being 100% solution, if we've got a 30% solute solution, we're going to have a 70% water, right? What are we going to have over here? We're going to have a 50% water solution because a lot of it's solute. So where is the water going to go? Out of the cell. Okay? Because water's always going to move from its higher concentration to its lower concentration. Okay? So what do hypertonic solutions tend to do to cells? Suck the water out of them, right? In your lab handout, it very indelicately states, salt sucks. Okay, salt draws water out of other solutions. Okay? What if we reverse this? What if we put a cell with a whole lot, well, let's leave this at 30%. What if we put this in a solution now that's only 10% solid? Or 90% water? Is this hypotonic or hypertonic compared to the cell? Okay, this is now hypotonic. It's got less solute than the cell does. Okay? So is the water directional movement going to be the same? No. Now we're going to have the water going from its higher concentration to its lower concentration. And so water's going to enter the cell. And the cell's going to swell. So do you see how the status of the environment in terms of dissolved solute is going to have a real consequence to the cell in terms of whether it gains water or loses water? What if we stick it in a 30% solution? Is that hypertonic? Is it hypotonic? No, it's isotonic, right? Will water enter the cell? Yeah. But it'll leave the cell at the same rate, right? So it'll be in equilibrium. If you have a really bad injury and you have a lot of blood volume loss, one of the things that they need to do is they need to kind of restore your blood volume. And if they can't give you a transfusion for some reason, maybe there's not blood available, they can at least put fluid in there. And they can give you a fluid bolus to try to re-up your amount of fluid in your circulatory system. Should they put pure water in as a fluid replacement? No, they shouldn't, because there's always some dissolved solute in the cell. And pure water is 100%. So the cell will always be somewhat hypertonic compared to a pure water solution. So what would happen? Water would start rushing into your red blood cells, and they would swell, and they would lyse. So what do you want to do for fluid replacement if you have to give something? Get isotonic saline. Okay, saline that's got the same solute concentration of the approximate cytoplasm of the cell. And then that way the solutions will be isotonic to one another, and you won't have water gain or loss from those particular cells. Now, there's an important assumption that we're making when we give you this particular scenario. And that is that this membrane is impermeable to the solute, but permeable to water. If this membrane were freely permeable to the solute, would the solute maintain its concentration of 30% in the cell? No, it would start diffusing out to a lower solute concentration. There's zero salt out here right now. So if the membrane is differentially permeable to the solute, then we only have to consider the movement of water in response to water's concentration gradient. If the membrane's permeable to the solute, then everything is going to eventually equalize. So, here's a good practical example. We have this bent glass tube, which has a little membrane inserted in the bottom of the bend of this particular tube. We have a lower concentration of sugar molecules on this side of the cell, on the membrane, and a larger concentration over here. Initially, when we set this up, the fluid level of the two sides of the tube are equal. But what's going to happen? If this membrane is impermeable to the solute, then the solute's not going to equalize, and there's always going to be more on this side than this side. So this will be a hypertonic solution compared to this one. So what's going to happen? Water's going to move by osmosis from this side over to this side, and the water level will rise over time. Okay? But if this membrane is permeable to the sugar molecules, then they'll equilibrate, and then the water will be at an equal level on both sides as well. So, you're going to do an experiment that looks something like this in lab next week, where you're going to put an artificial cell, which is really kind of a piece of cellophane called dialysis tubing, and you're going to put some things in the cell and other things in the beaker, which represents the environment. And then you will make some observations about how things move. How are they going to move? They're going to move in response to differences in concentration gradients. And whether they move or not across that membrane will be determined by the permeability of the membrane to that particular molecule. And you can make some observations and some predictions based upon these kind of experiments about what the membrane is permeable to and what it isn't permeable to by observing how big or small the bag gets because of water gain or water loss, and also by doing some analysis like a Biuret test to see what's happening to the protein, or a Benedict's test to see what's happening to reducing sugars, and you can kind of follow what happens to the movement of molecules across this membrane during the course of a very short period of time. You can set up an experiment like this, and in about half an hour, you can get some pretty meaningful results. So, plant and animal cells respond to the solutions that they are placed in. The response is a little bit different depending upon whether it is a plant cell or an animal cell. Here's what happens when you put an animal cell in hypotonic, isotonic, and hypertonic solutions. And here is, by contrast, what happens to a plant cell. What's the difference? The big difference in response is due to the presence of the cell wall in the plant and the absence of the cell wall in the animal cell. So if we put an animal cell or a plant cell in a hypotonic solution, what's going to happen? Water's going to leave the environment and go into the cell, and eventually enough turgor pressure will build up in the cell to result in cell lysis. This is what would happen if you replaced your blood with pure water in a medical emergency. The red blood cells would pop and lyse because that water would be hypotonic compared to the inside of the cells. Plant cells will also swell, but because of the restraining cell wall, they're going to get nice and crisp, or another word for that is turgid. They're swollen. And that's actually kind of ideal for a plant cell. Because if you're a leaf cell, for example, and you're nice and swollen and turgid, then the leaf can have some structural integrity, and it can be out there and have a nice surface area to absorb sunlight. What happens in an isotonic solution? We get an equilibrium. Okay? So the red blood cells have this normal kind of biconcave disc shape, In a plant cell, they get a little bit wilted or flaccid because there's not a buildup of internal pressure here. They're not swollen. So plant cells, even in isotonic solutions, start to wither a little bit, and tissue starts to wilt a little bit. If we put animal cells in a hypertonic solution, a very salty solution, they're going to shrivel. And what you'll see in a plant cell is that the membrane of the cell will pull away from the cell wall. And you can actually watch this happen if you take plant tissue and look at it under the microscope and add a very salty solution to it. You'll see the cytoplasm of the cell all draw in toward the center of the cell. As the turgor pressure is relieved because water's rushing out, and this particular state where everything kind of shrivels up into the center of the cell is called plasmolysis. Obviously, this isn't good for the cell, and this isn't good for the plant tissue that the cell is part of. Once it gets to be spring, one of the things you'll start to notice as you're driving, particularly along maintained roads like the interstate or some of the county roads and things right here, is if you look off to the side of the road, a lot of the plants that are emerging look all shriveled and almost burned. Anybody know why? Okay.
Salt they put on the roads.
Salt they put on the roads, okay? The salt creates a hypertonic environment and literally sucks the water out of the roots of the plant. Normally, we want the water to be going from the soil into the roots. But we reversed the situation by sucking the water out of the plants. You can also do that if you over-fertilize plants. Sometimes people will tell you, be careful with the fertilizer, you'll burn the plants. You're not really burning them. Fertilizer is solute. It's things like phosphate and nitrate. But it creates a hypertonic soil solution, and it pulls the water out of the plants and causes those tissues to plasmolize. And that causes the plant tissue to die, and it kind of looks like the plant has been burned. Animal cells and plant cells face different challenges because of the absence of the cell wall in an animal cell. And paramecium, for example, that live in hypotonic environments are constantly challenged with ways to keep from exploding because they're constantly gaining water. So what do? They have adaptations like contractile vacuoles, pump the water out as it enters. That particular type of mechanism is a type of mechanism called osmolar regulation. And contractile vacuoles are one strategy to achieve osmolar regulation. Plant cells don't quite have that same pressure because of the presence of their cell wall. So really, in some respects, the plant cell wall is kind of an adaptation that means they really don't have to have these other osmolar regulatory mechanisms. So plants and animals face different selective pressures. Remember that term from last semester, evolution? Based upon the fact that they are different structurally. Okay, so there's our filling vacuole within the paramecium, and then that migrates to the surface of the membrane and discharges the water. And so here is a contracting vacuole that has pumped some water out. So that's what a couple of the experiments that we're going to do on Monday are going to be doing, is reinforcing some of these ideas of what happens to cells or models of cells when we put them in different solutions. The other thing that we're going to do some experiments with is investigating some phenomena that involve traffic of substances across cell membranes. I'm not going to go through all of this. This is another one of those outline slides that kind of give you an idea of what we've talked about already and what we're going to be talking about in this particular section. I think those particular slides can be useful summaries when you're studying. So we're going to talk about two scenarios. We're going to talk about, first, how do smaller molecules move across membranes? And then after we go through that in a fair amount of detail, we're going to talk about how some larger molecules move across membranes. So let's take the small molecules first and talk about those a little bit. We said the small molecules move by two general processes. One is passive transport, which is diffusion across a membrane. There are two types of passive transport. There is simple diffusion, and there is facilitated diffusion. What's the same in both of those? The direction of movement. Because this is diffusion, things are always moving from higher concentration to lower concentration. The difference in these two phenomena is that in simple diffusion, the membrane is permeable to the substance diffusing. So what kind of substance might it be? Might be very small. Probably is nonpolar. Because those are the kind of things that membranes let across very easily. So oxygen, carbon dioxide, those gases, can freely diffuse across the membrane, and their movement is governed just by where's the concentration highest and where's it lowest. And things move from high concentration to low concentration. This is spontaneous, so it doesn't require the expenditure of energy. In facilitated diffusion, the membrane isn't permeable to the substance. It might be too big. The substance might be polar in nature. But there's something about it that prevents it from getting across that membrane. So let's say right here that this part of the aisle represents the membrane. And you're sitting in the cytoplasm. And I'm out here in the environment. Okay? If the membrane is permeable to me, it's like I can swim across this body of water. I have the ability to move across it. If the membrane is impermeable, I'm not a swimmer. I'm going to drown when I try to go across that body of water. What's an alternative if you're not a swimmer? Have a raft. Okay? And the raft in this particular case we sometimes call a carrier molecule. It's a transporter. And what kind of molecules are the transporters? Are they lipids? No, this river, and we haven't talked about really the structure of the cell membrane yet, but this river is the lipid portion of the membrane. What kind of molecules are the rafts? What's the other major component of cell membranes? Phospholipids, and they're proteins. Okay? And these carriers have specificity. Only certain things can get on the raft. Other things the raft won't pick up. Proteins that have specificity. Where have we heard that before? I've actually heard it in two contexts now. Remember, one was antibodies. Antibodies react with specific foreign antigens. And another instance we're going to talk about in a fair amount of detail when we talk about metabolism is enzymes. Proteins that have specificity. So these carrier molecules in some respect behave a lot in the same way that enzymes do. Things are still moving from high concentration to low concentration. No energy is required for facilitated diffusion. The facilitated refers to the fact that it needs something to get across that impermeable membrane. It needs help. It needs an assist. So, here are your passive transport phenomena. Here is simple diffusion. Here's our membrane right here. These are phospholipid molecules that make up the core of the membrane. This is a hydrophobic environment inside. So if molecules are small and nonpolar, they can pretty much just move through that membrane on their own. Which direction are they going to go? High concentration to low concentration. Simple diffusion. Here, these molecules, there's something about them that the membrane doesn't like. It won't let them across. Maybe they're too polar, which makes them hydrophilic. Maybe they're a little bit too big. Glucose is a fairly big molecule. It has difficulty moving across this phospholipid bilayer. But do we want to get glucose into the cell? Absolutely. Okay. We need glucose as a fuel for what? The process uses glucose in the cell. Cell respiration, right? We can make our ATP. So if you eat a big meal and it's got a lot of carbohydrate, and you gobble that up, and that carbohydrate passes through your stomach into your intestines. There it starts encountering some enzymes that start breaking that polysaccharide down into the glucose units. And now you want to get it from the cavity or the lumen of your intestine into the blood. Well, what's blocking it? The epithelial cells that line your intestine. And they can't cross the membrane because glucose is polar, it's a little bit big. So even though there's a whole bunch of it sitting in your intestine and the blood sugar levels in your blood are dropping, it's not going to cross that barrier without some help. So it's going to need an assist. It's going to need some help, some facilitation. This is a couple of representations of how the protein might assist the transport across that hydrophobic river, if you like. This particular arrangement is called a channel protein. And basically what it does is it creates kind of a tube in the membrane. The inside of this protein is lined with amino acids that are hydrophilic. And therefore, polar substances are happy inside that tube because that's a hydrophilic environment. But the protein folds up in such a way that these other amino acids that are out here facing the membrane are hydrophobic amino acids. So that particular molecule folds up so it's got, non-polar amino acids out here and polar amino acids in here. And this creates a channel that these can move through. Once this channel is established, it moves from high concentration to low concentration. Another strategy that is typically used in these carrier molecules involves conformational change. Remember that term? We introduced the idea of conformational change when we talked about flagella and cilia moving back and forth. And that dynein walking, remember that? Those red protein molecules that flipped from one shape to another. Remember what caused them to flip? Whether they were phosphorylated or dephosphorylated. Remember those terms? Phosphorylation, a phosphate group gets stuck onto that protein, and what does that do? It changes its shape. What happens when it loses the phosphate? It snaps back. Okay? So these proteins change shape depending upon whether they're phosphorylated or dephosphorylated. Okay, so what if I've got a carrier molecule and it's facing the outside of the membrane with this particular shape? And this represents a binding site. This represents a place where glucose can transfer, can attach, sorry. And it binds to it. What does that do? It causes a conformational change, or a change in shape. And what might that do to that carrier? Flip it the other way. So the carrier basically rotates from one side of the membrane to another based upon whether the glucose is attached. And then once it flips over to the other side, there's low concentration of glucose here, it's going to be released. And when the glucose is released, what does the carrier do? Flips back again. So it flips back and forth from side to side based upon whether the molecule it's carrying attaches to it or is released. And that's going to be really important in a minute when we talk about another type of transport called active transport. That idea of conformational change and phosphorylation and dephosphorylation is going to be really important. Okay, so this is, you can see the example a little bit better here. So this is facilitated diffusion. Here is your carrier molecule in this particular conformation or shape. And when the transported substance binds to it, it triggers a conformational change. It swivels so that the other side becomes open. And this particular binding site no longer will hold the solute, and it'll be released. Once it's released, it snaps back here again. Does that require energy? No. Okay? It is just driven by the fact that molecules binding to proteins can cause their shape to change. Now, if these were phosphate groups that were produced as a result of ATP hydrolysis, that would be an energy requiring process. But if this is just something like a glucose molecule that needs to get from here to here, that's not energy requiring. Okay, active transport. This one's going to be a little bit challenging. This one's quite important, particularly in your bodies. One of the things that we'll see is that your whole nervous system and its function depends upon this phenomena of active transport. The functioning of nerve cells and the propagation of a nervous impulse is really the product of ions flowing across those neuron membranes. And the ions that move are sodium ions and potassium ions. Are membranes normally permeable to sodium and potassium? Nope. They're small, right? But why aren't the membranes permeable? They're charged, okay? They're very hydrophilic. When a nerve cell gets stimulated, an impulse is propagated along the membrane of that neuron. And that membrane propagation is the result of sodium ions flowing one direction and potassium ions flowing another direction. And what happens after that nerve is stimulated? It's got to recover, right? And the sodium ions and potassium ions have to move back across the membrane and kind of reestablish their normal situation. And then when they're stimulated again, they start moving. And then they get recover again, and they have to reestablish. That reestablishment of the membrane potential is dependent upon this phenomena called active transport. So if active transport wasn't working properly, your nervous system wouldn't work. And if the nervous system doesn't work, you don't work. Okay? So, what's happening here? Active transport involves the pumping of solutes against their gradients. So where are things moving? They're moving from low to high concentration. That's not the same as passive transport, is it? passive transport is spontaneous. And that means things are moving downhill from high to low. In active transport, things are moving from low to high. If you have a boulder at the top of the hill, once you give it a little nudge, it rolls down it on its own. That's spontaneous. Does it spontaneously roll up the hill? No, we've got to expend energy. And that's what's happening in active transport. We're pumping things against their gradient up the hill. And it requires energy, and that energy is provided by ATP hydrolysis. PI stands for phosphate. And so basically, adenosine triphosphate, you lop off a phosphate and you're left with adenosine diphosphate. Okay. So the membrane's impermeable to these things that are being moved. So we need a carrier. We need a protein involved. And things are moving from low concentration to high concentration, so energy is required. This is not a spontaneous process. Sodium potassium pumps are important, and actually plants sometimes, oftentimes, will actively transport nutrients from the soil into their root cells. What would be the advantage of doing that? Why might a plant want to actively transport, at the expense of energy, solute from the soil into its root cells? What would that do for it? What kind of environment would that create inside the root cells? It accumulated more solute. Hypertonic environment, a more salty environment. And what would salt water do? It would follow that solute. And so that would facilitate sucking water out of the soil into the plant. Okay? Just the opposite of what happens when you put all the salt on the soil. You accumulate the solute by active transport in the roots, and then the water will follow. Okay? So that's an intentional, if you like, strategy that plants use to keep water flowing out of the soil into the roots. So simplistically illustrated, in active transport, what's happening is these particular substances are being moved from a lower concentration outside the cell to a higher concentration inside the cell. Because that's going against the concentration gradient or uphill, it's going to be an energy requiring process. And we need this carrier molecule to facilitate this because the membrane isn't normally permeable to these substances. Now, as it turns out in active transport, there's an actual exchange that's going on. Some things are being moved into the cell, and other things are being moved out of the cell. And that's definitely the case in nerve cells. After they've been stimulated and they have to regain their resting potential, some ions go back outside the cell, some come back inside the cell. Okay, so here's a representation of how the sodium-potassium pump works in the membrane of a cell. It looks a little complicated. It is a little complicated. And before we dig into it, there's some kind of general things that I need to point out to you about this sodium-potassium pump. This particular pump is a protein that's called a sodium-potassium ATPase. What does this tell you about this protein? It's an enzyme. And enzymes catalyze reactions, right? They speed up chemical reactions. What reaction does this particular enzyme catalyze? The hydrolysis or breakdown of ATP. Sometimes this enzyme is active and does this, but other times it's inactive. What's going to determine whether that enzyme is active or inactive? The shape of the protein, its conformation. What's going to change its conformation? Conformations will change depending upon what molecules are binding to that protein. So that's something we're going to have to follow and kind of consider. This protein is also a carrier. And it has 3 binding sites for sodium atoms. It has 2 potassium binding sites. And it has 1 phosphate binding site. So all kinds of different molecules can attach to this protein. And what happens when they attach? It's going to cause a shape change in the protein. So these binding sites are also active, inactive. Meaning sometimes they'll bind something and sometimes they won't. What does that depend upon? The shape of the protein. So you got a lot of weird things going on here in terms of this molecule changing shape in response to something binding or something releasing. And what's the consequence of those shapes changing? Maybe the enzyme becomes active, or maybe it becomes inactive. Maybe it binds sodium or releases it. Or it binds potassium and releases it. Or it binds phosphate groups and releases it. The net result is that if the pump operates properly, through every cycle that it goes through, it pumps 3 sodiums out of the cell and 2 potassiums in. So if this is a nerve cell, when it goes through the recovery process after it's been stimulated, it pumps sodium back out here and accumulates potassium in here. And then what happens next time that nerve is stimulated and an impulse is propagated? Potassium runs this way, sodium runs this way. And then it's got to recover. It's got to pump these out and these back in again. So it's constantly going through this cycle of stimulation recovery. And in order for that nerve to keep functioning, that pump has got to keep reestablishing the proper levels of sodium and potassium inside and outside the cell. So all we have to do next time is kind of follow through and see kind of what's the cycle of events. What happens when sodium binds? What happens when potassium binds? What happens when phosphate binds? Okay? So this is one of the more kind of conceptually difficult parts of this material. So we'll make sure we go through that again on Tuesday. Have a good look at it. Make sure you read your lab.